Hello, my name is Grissel Trujillo de Santiago, and today I'll talk to you about continuous chaotic printing to fabricate multi-layer micro and nanostructures. This is our wonderful group here in Mexico, and we are dreamers and we are very ambitious. And I'm sure that we share this dream with many of you. We want to enable the bioprinting of full functional organs. And of course, there are many challenges to overcome, but we as a research group want to focus on the microarchitecture. Our tissues are not homogeneous masses of only one type of material or cell, but they have this fine microarchitecture that is composed of multiple materials, of multiple cells, and this is closely related to the biofunctional functionality of the tissue or organ at the macro scale. So I hope to convince you today that chaotic 3D bioprinting is a very practical and cost-effective tool to face the challenges. I like to use this example to explain chaotic printing. When you pour cream in your coffee, very often you see these structures forming. This happens because the flow is drawing, if you allow me, those structures. Now, instead of using cream and coffee, we can use cells and hydrogels. So the cells can create these structures within a liquid pre-gel, and then I can apply a cross-linking stimulus, for example, light, and hold or freeze the structure. A problem with this example is that the process is not reproducible. And well, that's why we use chaotic mixing. I know it sounds counterintuitive, but I'm talking about deterministic chaos, which has nothing to do with turbulence and has nothing to do with disorder. This is the chaos of the dynamical systems theory. So these are some structures that chaos can make. There are many chaotic systems. So far, we have reported the use of only 2, but we are currently working in the lab with 5 more. And what I want you to see here is that these structures are multi-layered and that we can make them multi-material. Fortunately, our tissues exhibit very similar structures. For example, one layer of one type of cell will share interface with a layer of another type of cell or material. And well, these are some key characteristics of chaotic flows. Of course, they can create a complex structure, as you already saw, and they are deterministic. You can model them using Navier-Stokes equations, and this can make them reproducible, predictable, and well, actually here you can see a structure made by Carlos and Joanna in the lab using a chaotic flow, and it's matched with the computational simulation that Diego made. Another very cool characteristic about chaotic flows is that they grow exponentially. This means that we can create a structure very efficiently. A chaotic mixing recipe will iteratively stretch and fold a fluid, while a regular mixing recipe will only stretch the fluid. The stretch and folding actions is what makes chaos exponential and so effective in creating a structure very fast. And that's actually why people use this methodology to homogenize in the laminar regime. But no one was capitalizing chaos for manufacturing multi-layered constructs. And here I'm showing you the Kenix static mixer. You can think about it as a bioprinting head. These are the mixing elements. These are static and are housed in a pipe. They are the ones that do the magic, these mixing elements. When you perfuse your bioinks through the Kenix, the mixing elements reorient and split the fluid in every single element. At the outlet, you will have this layer microstructure hydrogel filament with intercalated layers of materials that the materials that you fed. This is a cross section of our extruded filament, and this is a computational simulation of the flow. Also, you can determine how many layers you want within your filament just by using more or less mixing elements. For example, one element renders 2 layers, 2 elements renders 4 layers, 3 elements 8, 4 16, 5 32, and so on and so forth. Here, it's also very clear that the process is exponential. These are actually computational simulations, and these are experimental images that match with the simulations. So you can anticipate that the idea is to use this as a bioprinting head in a regular extrusion bioprinter. So we can we can enable the bioprinting of three-dimensional constructs at the macro scale, but with control at the micro scale, which is key again in tissue functionality. Also, I want to elaborate much on this, but we can couple our static mixer to an electrospinning setup. And when we use the proper blends of polymers and the right process conditions, we can produce electrospoon fibers with inner nanolayers. As shown in these images, obtained by photo-induced microscopy that allow us to see, that we effectively preserved the layered structures created in the kinetic static mixer all the way to the nanoscale. Okay, going back to the microstructure, the microscale, we wanted to print something alive and to assess the resolution of our system. So we first choose bacteria, E. coli, because they are very small, only 2 microns in average. And we were capable of printing hydrogel filaments with 1 millimeter diameter with up to 7 Kenix mixing elements. And this render 128 internal layers, and each one of them had a thickness of 8 microns. This is awesome because this resolution is not achievable by any other bioprinting, extrusion bioprinting technique today, as far as we know. Next, Carlos and Joanna printed muscle precursor C2C12 cells that are murine myoblasts, and the cells were loaded in a GELMA alginate hydrogel, and these other layers are pristine alginate. The rationale of this architecture was to provide barriers for the cells so they couldn't cross barriers, and this could allow them to align, to promote cell alignment lengthwise. Here, we show that the process is cyto-friendly. This is a live and dead assay to show that during 28 days of culture, we have a high viability. And a very cool advantage of our technique is that we can create a lot of thin layers, or said in a different way, we can achieve a very high resolution without paying a high cost in terms of shear stress. Here we are showing the velocity field along the mixer, and we see basically the same velocity values and therefore shear stresses values along the pipe. With conventional extrusion bioprinting, high resolution requires narrower outlets and therefore higher shear forces that will affect cell viability. And it does not happen in our case. Well, these are macrographs after 18 days of culture. This is the ghost of the filament as a reference. But here you can see that the myoblasts merged into multinucleated cells and that they preserve the position in which they were initially printed. And also you can see that over time they align to the main direction of the fiber. This was assessed by image analysis by measuring the orientation of the active fibers. We also did immunostaining to reveal the presence of alpha sarcomeric actin, a relevant biomarker for muscle microarchitecture. It was there. And also we measured the alignment within these micrometric structures. And again, we saw a very high orientation. So cool. This means that the process is self-friendly enough to allow tissue maturation, or at least some signs of tissue maturation. We can adapt this process to make it multi-material. So far, I showed you scenarios with 2 different materials, but nothing prevents us to do more. For example, here, Carlos and Joanna adapted 4 inlets in the lid, so we can perfuse 4 different inks. With this, we can pre-design patterns too. For example, if you want a pattern like this one, orange, black, green, I can use this setup in the inlets. And this is very cool because the pattern remains no matter how many Kenix elements I use in my bioprinting head. So see that here is again the pattern: orange, black, green, orange, black, green, orange, black, green. Okay. And well, we keep pushing the limits. We have been capable of, with collaboration with Trike, to print up to 8 different inks. Lastly, we can adapt our system to make refusible hydrogel filaments. We can use a combo of a permanent and a sacrificial material to create filaments with hollow microchannels. Here, we use gamma alginate as a permanent and pluronic as a sacrificial. Here, Carlos and Joanna were capable of challenging the system to print these multi-channel filaments with 6 elements, again, using gamma alginate and pluronic. Now we are working on loading cells in these filaments in the permanent hydrogels so we can create vascularized like tissues. So with this, I hope that I have conveyed the message that chaotic printing is a very simple and cost-effective tool capable of very elegantly help us to overcome these important challenges in biofabrications and hopefully can put us closer to achieve, well, this ambitious dream. I close acknowledging my students, mentors, and collaborators involved in this project to my institution and funding agencies. Picture of the last picture of the group in Zoom. And well, thank you very much. Bueno, voy